So thank you again, and thank you very much for the invitation. And I've broken the talk up into a few short parts. I'm not going to talk about new treatments in great depth because I've got a whole 12 minutes to speak. So what I thought I'd do is talk a little about DNDi for those who are not familiar, and then focus on just a couple of examples of lessons that we've learned in the drug discovery, drug research process in the last half dozen years. One example with Leishmaniasis on the chemical diversity, and one with Chagas on disease models. And then probably just one challenge that I see that was alluded to a little this morning earlier in the panel discussion. So, there we go. Is the button working? Yep. So DNDi is a product development partnership. We're one of many, as you can see on the screen here. And PDPs work in different areas, all based, the product is the thing in common. We're all looking at making products, be they diagnostics, vaccines. We work in different areas. DNDi at the moment in neglected tropical diseases, and a mini portfolio, as we call it, in pediatric formulation for antiretrovirals. We all, as I say, we all have the same focus of products, so I think if you met with the different ones, we all have very, very different personalities. And DNDi vision is we are collaborative. We look to building partnerships. We're non-profit. We're largely virtual in as much as we don't have our own laboratories or research centres as such, though we do manage most of the clinical research through DNDi and through people working with DNDi. At the moment, we're focused on most neglected communicable diseases, and we have the rather ambitious objective of 11 to 13 new treatments by 2018. And I emphasize the word there, treatment. That will be in our first 15 years. If we get one new chemical entity in that period, I think, well, we're very hopeful that we will get one, possibly two, but if we do that, we'll be very pleased with our success. So we're not looking at NCEs in that period. But behind that, we are building a pipeline. So in the coming five to 10 years after that, we will start to bring new treatments, new chemical entities to patients. And we also work and strengthen, harness research capacity in disease-endemic countries, and that now spans from screening and early drug research all the way through to phase 4 trials. So just one more slide before I get a little bit onto the other topics. We have offices, we have several founding partners, really grew out of Médecins Sans Frontières. And we have offices around the world, and last year, just after our 10th anniversary, was the first time that we employed more people outside of headquarters in Geneva and in the regional offices. And the major offices would be in Latin America, in Rio, in Kenya and Kinshasa, and Nairobi and Kinshasa, and in Delhi in India. And as DNDi, I don't think it'll grow very rapidly from now, but what growth we do have, we aim to do in the regional offices where the diseases are endemic. I should point out we have offices in the north in the US and Japan, but that's largely for liaison and fundraising. In addition to the 140-odd people now that work with DNDi, work for DNDi, we have about 4 to 1 people who work with us as full-time employees on contract research in different areas. And again, the vast majority of those are in countries where the diseases are endemic, particularly at the moment in sub-Saharan Africa. And I won't go through the pipeline in any detail, except to highlight that in the middle part here, in particularly, or DNDi, we have full programs for Leishmaniasis, Chagas, and we've stopped research recently in African trypanosomiasis. And I should point out here in Chagas and Leishmaniasis, the pipeline is very bare. We are starting now to generate some preclinical candidates here, as are other groups around the world, in particular University of Dundee and GSK. But we don't have much that's moving forward in the next year or so into translation. And so what I'll talk about, rather than go into that, is to discuss just a couple of issues that we've identified in research. And the first is the paucity or the lack of chemical diversity. And this is a case particularly in Leishmaniasis. And largely the screening that we've done, and I should say now, is phenotypic screening, where we're looking to hit the A mastigotes in a host cell. So we're looking at the hit getting across a couple of membranes through a pH gradient and across the membrane of the parasite into hitting the target, which is quite a tall ask for any molecule. One approach is to look at this is increase screening capacity, and that has been done. And I think Julia will be talking a little about that later this morning, this afternoon. and the efforts of GSK, but I would say probably 3 million compounds collectively in Pasteur and Korea and Dundee and GSK have been looked at for Leishmaniasis, and we still don't have a huge number of lead series or hit series even that we want to take forward. So a couple of approaches we're looking at. One is encouraging. We're not, unfortunately, haven't got the money to fund target deconvolution, but each hit most hits, I should say, will attack a chemically validated target. The only problem we have is we don't know what that target is. So if we can identify those targets, then we can get into some target-based drug design. And one that we're working on, and I won't go any further because I'm sure Wes will talk about this as well, is the pathogen box, and we're collaborating with MMV there to make compounds available so researchers can start to do some of this target-based research with hits that have got a decent amount of annotation behind them. Another one, and this is stepping a little outside my expertise, but is looking at computational chemistry approaches, and this is some work that Jean Berriesse did at DNDi a few years ago, looking at the actives that we have and putting them back into the library to see if we could look at structural variations, modifications to come up with new series. And out of that, did come up with a number of new clusters and brought some hits through to hit to lead. Another one I'll spend a bit more time on, just a few minutes, is a project that was brought forward by Charlie Mowbray at DNDi and called the Drug Booster. I should say Charlie originally called it pre-competitive research, but our communications group thought that wasn't emphatic enough. But basically, it's an approach at multilateral collaboration. So most of them, in fact, all of our collaborations to date with pharma have been bilateral, and this is a multilateral collaboration between DNDi and pharma. And the principle is that, and I won't go into it in great detail, but we start here with a seed compound. It's a validated hit, and that can really come from anywhere, from any of our screenings or any of the pharma partners. And we give that to a number of different pharmaceutical partners to take back and screen in their library to look for analogues. And this has actually started now. We haven't actually announced it yet. I think there's a few things to tie up, but we have now 4 pharma online with this program. And those pharma partners will screen their libraries in silico and come up with 100 analogues using their own algorithms for screening in their own libraries and give that back to DNDi to screen. And out of that, we take one improved hit plus the SAR, the structural activity relationship around those 400, in this case, compounds, and we put that back into the system and feed that back in and do that for 2 to 3 iterations. And out at the end here will come the best hit series that we have, along with a great deal of annotation around that. The goal is, and it will depend on the agreements we have with companies, is to put that into the public domain. So the best of, I suppose, possibilities is to get that into the public domain, and from there we can take that forward independently or with others into lead optimization. And for us, the companies have come on board because for the individual collaborations it's quite small. They simply have to do an in silico screen. We're only asking for 100 compounds per screen, and we're only asking for 1 to come out in the public domain. So for the companies, there are a couple of levels of encumbrance searching, but in the end, we're not asking a great deal to be coming out into the public domain. So I'll move now to disease models. 5 minutes, yep. And Chagas disease in particular for this one. And it's to highlight some learnings that we had from a clinical trial we did a couple of years ago with an antifungal triazole, E1224, which was a prodrug of rabuconazole. We did this in collaboration with ETSI. And I'll quickly take you through this, but it was a trial where we had 3 doses of E1224, 3 regimens, I should say, and the control treatment benznidazole, the standard of care. What we did was after treatment around here was to measure by PCR, qPCR, the parasite in the patient's blood. And quickly I'll go through the black at the top here is the placebo control, and you can see that that was measured throughout the study. All of the other arms, both 1224 and benznidazole, went to baseline at the end of treatment. What you can see here is the green triangles they are of benznidazole stayed, or stars, stayed on baseline in essence, but all 3 arms of the E1224 came back up, and all patients had parasitemia up to about 12 months afterwards. So in a sense, it was a quite spectacular failure in that sense. From our perspective, though, it was a curiosity because 1224 is about 1,000-fold more potent than benznidazole. And in the publications of studies that had been done, it had worked in the animal models of disease. So we had to look back and, from preclinical research perspective, and try and find out what went wrong. I've just one slide to show you here, and this is looking at killing curves. And here, from now on, I'll talk about posaconazole because that was used. It's equally potent to benznidazole, same mechanism of action, and can largely be interchanged. And same class. So benznidazole here, you look with a number of different strains of T. cruzi, and you have a standard killing curve here, quite steep slope. And you see, once you get past the IC99 or MIC, you've got 100%, basically 100% of the parasites are killed. But if you actually count the parasites, and this is counting the individual amastigotes in the host cells, if you do that with posaconazole or 1224, you can look at one strain here, this 2-element strain, where it looks very similar, but not the same as benznidazole. With other strains, you'll find that you get this killing curve, but then it flattens out well before you get to killing of 100% of parasites. So there are residual parasites in the cell which are recalcitrant to posaconazole 1224. And you can see we've moved the IC50s here way to the left, but still, and you can go for logs above the MIC and the parasites are still recalcitrant. So within that cell, there are parasites which basically aren't hit by the drug candidate. So we can now do that in high content, medium throughput, at least automated screening. And we took most of the hits that we had, or all of the hits we had through those high contour, through this screening. And this really accounts to a large degree for such a paucity of hits in Chagas now, is that we killed most of them in doing so. But at least we can now do that in the primary screen instead of going to the clinic and discovering it then. And similarly working, and most of that work I should say was by Lucio Freitas at Unicamp in Brazil, in Sao Paulo. We've also worked with John Kelly at London School, and he's developed a couple of bioluminescent imaging models, or imaging model for Chagas in mice. And there's a paper which has just been submitted which now shows that posaconazole shows the same outcome in the murine model now as we saw in the clinic. So we now, pardon me, we can now go back to our models and hopefully eliminate or weed out these leads and hits before we get to the patient. So finally, I just talk about one future challenge, and this was alluded to or talked about a little in the earlier session. And that is, once again, moving towards elimination, eradication, particularly with the roadmap for implementation with WHO and the London Declaration, of which DNDi is part. And the question is, what lessons have we learned from the last time we talked about elimination and eradication in the 60s and 70s? And really, there are a couple of things I'd point out. And one is to read very closely the roadmap. Elimination of Leishmaniasis in India, which is ambitious in itself, is not elimination of visceral leishmaniasis in Africa, where the same drugs don't work, or in Latin America. So we're not looking at as grand a plan as is sometimes as proposed. And secondly, where those elimination programs are proposed, I think one of the big learnings is that last time it was seen as a very centralized program where leadership could be done from a central place and all situations were the same, and one size fitted all. And so we really do need leadership from endemic countries. We need to look at each region of endemicity as a separate problem and look at specific tools that we need, and those tools are not just drugs, but vector control, as was mentioned this morning, diagnostics and so forth, everything involved in eliminating or treating that disease, as well as looking to overcome many of the IP and regulatory barriers. If we don't, I think R&D will suffer because people will stop funding or reduce funding in R&D because of the promise of elimination in a very, very short period of time. On that, thank you very much.